first step in purifying protein is to actually generate the protein that you're ultimately trying to purify. For this course, we're trying to purify an alpha-galactosidase, which we've grown recombinantly on an overexpression plasmid in E. coli. We've grown this E. coli in an autoinduction media in order to maximize the amount of alpha-galactosidase produced. So the first stage, once you've actually generated the cell biomass itself, is to extract the cell mass from the media solution as a whole. And we do this by centrifugation and resuspension in a buffer solution. So the first thing we'll do here is actually divide this cell culture up into these centrifuge bottles and spin them down to pellet the cells. It's important not to overfill these centrifuge bottles, because when they go in the centrifuge themselves, if you get any leakage during the centrifugation process, then you'll have to clean out the entire centrifuge. So we usually fill these to about halfway to 2/3 of the way to avoid any spillage issues. It's always important to make sure when you're centrifuging that your samples are balanced against each other. To do this for the large centrifuge, we try to make sure that our bottles are paired with bottles that are within 0.1 grams of each other. It's good practice to number the pairs of bottles going into the centrifuge so as not to mix up the bottles that pair together for their balances. To do this, we simply tare the first bottle, set it to 0, and then weigh the other bottle against it. In this case, this bottle is 0.18 grams heavier than its partner, so I'll remove mass from this one and place it in the other one to balance them out. Now we'll re-tare the first bottle. And check the second bottle against it. Now here I moved too much mass, and so I will add mass from the first bottle back into the second. Again, we'll re-tare the first bottle. And weigh the second against it. Now they show that they're equal mass, and so they can be paired together in the centrifuge. Now that we've balanced our samples, we'll actually load them into the centrifuge. It's important to always put the balanced pairs directly opposite each other in the centrifuge container. This centrifuge contains 6 slots, so we'll put each of our 3 pairs across from each other to load the centrifuge. Make sure the caps are on nice and snug so that there aren't any leakage concerns when you run the centrifuge. And then screw the top down tightly. We'll run this centrifuge at 6,050 RPM for about 10 minutes in order to pellet out the cells. Whenever you start a centrifuge, always make sure you watch it get all the way up to max speed to ensure that it's properly balanced when the run starts. Now that we've reached max speed, we'll leave the centrifuge to run for 10 minutes and then collect our samples. Now that we've centrifuged our samples, you can see we have a nice cell pellet at the bottom corner of each of these bottles. So for now, we'll just decant all of the supernatant liquid out of here, reserve the cell pellet, weigh it, and then resuspend that in the buffer that we actually want for our FPLC run. Now that we've weighed just the wet mass of the cell, we can calculate what volume of FPLC buffer to resuspend the cell pellet into. Typically what we do is about 5 mils of the buffer per gram of cell mass. Now for each of these centrifuge bottles here, we have somewhere between 2.5 to 5 grams of cell mass pelleted out. So I've done the calculations elsewhere to figure out what massive cells there are and what volume I'll be adding to them. And then it's just a simple resuspension. During this resuspension process, it's important to make sure that you pipette up and down around the cell pellet until the pellet fully goes into solution. This may take a little while, as the pellet is adhered pretty strongly to the sides of the bottle after centrifuging. But you can see that the liquid is starting to get a little cloudier as the cell mass goes back into solution. The buffer solution that we're using here is called IMAC-A, which will be the first buffer of the IMAC purification process. So we're just putting it into this buffer out of convenience for later processing. Depending on what kind of purification method you might be doing, you would choose a different buffer possibly, like AEC buffer A or HIC buffer A. Now that we have our cells resuspended in the buffer that we're going to run on the FPLC, we're going to actually lyse open the cells so that we can extract the protein inside of them. The method we're going to use for that is French press, which is this instrument right here. Now, in spite of the fact that it looks pretty chunky and, I'll be honest, it's going to make a pretty loud sound in a minute, it's actually one of the gentlest methods for breaking open cells and extracting the protein. Essentially, this system is just a very large pressure vessel, which is contained right here. There's 2 valves, an inlet valve on the back and an outlet valve on the front. And what we'll do is open the inlet valve, place the inlet tube in the back here into the sample, and then switch this ratio selector to down. So that'll actually start to open up the French press, and it'll draw our cells into the pressure cell right here. Then we'll close off that back inlet valve. We'll open up the outlet valve just a little bit, and we'll switch the ratio selector here from down to either medium or high. This will then start to compress the pressure cell and squeeze out our cell mass through this small outlet tube, where we'll have a collection vial waiting here. Now, I'm explaining this all in advance because once it's running, the hydraulic pump in here is going to be pretty loud and you won't be able to hear me. But that's essentially the process. So to get started, we'll first load up the sample into the pressure cell, switch the valves, change the ratio selection, and then collect the effluent from there. Now that our sample's loaded, we've closed the inlet valve. We'll get our collection tube. Open inlet valve closed, open the outlet valve, just a little bit so that we have a lot of back pressure here. and then switch the ratio to raise the pressure cells. It'll go fast at first as it squeezes out the air that's in the system. And now that we just have liquid in here, we want a very slow drip coming out to really maximize the pressure that the cells are feeling. Turn that up just a little bit. You can control this flow rate with the throttle of the outlet valve. And there we have our sample in the collection tubes. Now typically you'll run a sample through this process 2 to 3 times to really make sure you've lysed the cells well and are able to extract the protein from it afterwards. Now that we've lysed our samples, we have the crude cell extract we need to start the protein purification process. But the first thing we're going to do is take advantage of the fact that the protein that we're purifying from the E. coli extract is actually from a thermophile. And so it has very high thermostability compared to most of the proteins that are native to E. coli. So in order to get rid of some of that unwanted protein and really focus on the alpha-galactosidase, we're going to heat treat the cell extracts for a half hour at 65°C. And then centrifuge again in order to crash out the denatured protein that we don't want. Our thermophilic protein, though, which is thermostable, will stay in solution and will keep the supernatant from that centrifugation process and then use that reduced fraction of protein for the later protein purification steps.
Today we're going to show you how to buffer exchange proteins using these conical concentrators. The conical concentrators have filters that are affixed inside of these conicals that look like normal 50 ml conicals, except they have these filters in here. These filters have a set membrane size with a particular cutoff. This one is a 10,000 molecular weight cutoff, or 10 kilodalton cutoff. For today's demonstration, I put a blue colored dye inside of here, which will allow us to visualize the buffer exchanging process, where we will concentrate this down, add the new buffer to it, concentrate it down again, and repeat that process approximately 3 times. For high salt concentrations, you might need to use additional washing steps to get a full buffer exchange. But for today's purpose, we'll be able to visualize the passing of the dye through this filter and exchanging that with the non-dyed liquid. It's important to know what speeds to run your conicals at. Today we're going to run this at 4000 G in a swing bucket centrifuge. So I'm going to go ahead and centrifuge this to pass some of the liquid through before adding some of the water to start the buffer exchange. So now that the centrifuge is done running, we'll open it and take a look at what happened. As you can see, some of the liquid has passed through and some of it has remained on the top. So this is approximately 2.5 milliliters of liquid that passed through in a minute that we centrifuged it. So we'll actually need to centrifuge this further to get it to roughly half the volume before adding an equal amount of the liquid again. Now I've added water to the top part after passing the liquid through, which has diluted, and as you can see, it is less blue than it was before. So now we're going to centrifuge it again to concentrate it down further. So this is after the first round of buffer exchanging. As you can see here, this is the intensity of the dye that we started with, and in the top here you can see that the dye is diluting. As the liquid passes through, the dye accumulates in the bottom, as well as buffer that we've put into buffer exchange, which is why it is not as dark in the bottom as this as it is in the original sample. So we're going to go to a second round of buffer exchanging by adding water again to the top to dilute it further. So this is after the second round of buffer exchanging, and as you can see, the top is becoming clear as the dye passes through. Note that this dye is a very intense dye, and so even a little amount will make it appear a little bit blue, but it is becoming quite clear already. So we will go and do a third dilution, which will bring it to about a full buffer exchange.
So now that we've finished running our whole workflow on the FPLC columns, we're left to analyze how much protein do we actually have in those fractions, and how much of that protein is our alpha-gal. To answer that first question of how much protein is present in each of the fractions, we're going to use a Bradford assay to evaluate the protein concentration. So we have samples from each of the fractions we've run over the course of this class, starting from the non-heat treated cell lysate of E. coli all the way to the post-run fractions of the size exchange column. So from there, we'll take samples of these and run them on a 96 well plate using a Kamasi protein assay or Bradford assay stain. Essentially, what this assay does is interacts with the protein using this blue dye, and that interaction changes the color of the mixture from a brownish color to a bluish color, and we quantify that by measuring it against standards of known protein concentration. So we'll run a series of these standards at different protein concentrations on the same plate, and then use a linear regression of those standards to determine the concentration of our samples. One thing to note, you have to make sure that your protein sample is in the linear region of that standard curve that we'll make. So I've actually diluted all of the protein fractions that we're looking at by 100x to make sure that I'm in a low enough protein concentration that it'll show up in the right place on the standard curve.
Typical linear regions are between 0.01 mg per mil and 0.08 mg per mil of protein. Also note that our standards are made using bovine serum albumin, which is provided to us in a standard 2 mg per mil concentration.
The first thing I'll do to get started with the assay is to load the standards and my diluted samples, 20 microliters of each, onto the 96 well plate. I'll also load this plate in triplicate of every sample to make sure we have good statistical significance.
While Dan is loading that, I'll just explain a little bit more about the staining, how Kamasi blue, which is involved in this Bradford assay, stains the proteins. The Kamasi blue stain interacts with more generally with the amino acids in a protein, and is not that specific to one type of amino acid over the other. There are other types of protein assays, such as BCA, which react more specifically with certain types of amino acid residues and are less general staining. They work better for having complex mixtures of protein rather than specific purified protein, as they react with certain residues specifically. So in our case, where we have our bovine serum albumin as our standard and GalA as our experimental protein, if our stain reacted, say, specifically with tryptophan more, and one of those proteins has more tryptophan than the other, our calculations would be off because our standard would stain differently than our experimental protein. This is why we use Bradford reagent instead of an alternative staining procedure like BCA that would react specifically with those amino acid residues.
now that I've loaded all of the samples onto the 96 well plate, I'm going to add 180 microliters of the Bradford reagent to every well of this plate. Now, it is important to remember that the blue color of the assay develops over time. And so you want to try to add the reagent to your samples in as close to the same time as possible. So to help me out with this, I'm going to use a multi-channel pipette to load all the wells as quickly as possible and make sure that they all are exposed to the reagent for about the same time before I read the plate. So we'll pour some of the reagent into this boat here. And you can see it has that intense brown color that I mentioned before. And now, go sideways, right? Okay. I'll use the multi-channel pipette to grab a whole bunch of tips at once.
Note that you don't have to use all the ports on the multi-channel pipette at the same time to load. Notice how he is choosing them on the side. This allows us to conserve pipette tips so you don't have to fill the entire plate.
It also uses less Bradford reagent because I don't have to fill a whole bunch of empty wells in the process. It is important to be careful when using a multi-channel pipette to make sure that you're pipetting slowly enough that you get about the same volume in every single pipette on the multi-channel. There is some variability between them. Alright, and with that, the plate is loaded and ready to go. So you can see, as we're loading into the plate reader here, that the blue color has started to develop in some of our standards and in some of our samples as well. So now we'll analyze it at 595 nanometers to see what kind of protein concentration we have.
Hello, today I'm going to show you how to run an SDS-PAGE. So these are fractions from the GalA Purification on the final step using size exclusion, and I'm going to use that as an example today on how to screen fractions from the FPLC through SDS-PAGE, but generally it's to teach you on how SDS-PAGE works and why we do it. Specifically, SDS-PAGE is designed to denature proteins, to take apart any of that secondary, tertiary structures, to take it down to the base polypeptides, so we can determine their size on a gel. In order to do that, we have to take the protein samples that are currently not denatured and denature them. So the first step is we mix it with this compound called lamely buffer, which is a loading dye with SDS in it, which helps disrupt those intermolecular forces that hold the proteins together, as well as it has beta-mercaptoethanol in it, which helps disrupt the disulfide bonds in the proteins. And so, if you can see on the, I don't know if we can see this well on the camera or not, but this is an activity assay from the size exclusion run, and there are 7 fractions that had activity from GalA, and so we want to run an SDS-PAGE on that to determine the relative purity in order to tell what fractions to keep and pool together. And so we, as I said, we identified 7 fractions. So the first step is to take 10 microliters of your buffer, or the denaturing buffer, and add it to each of our PCR tubes. And then next, I will add the respective amount of protein from each of the collection vials into the respective tubes, also at 10 microliters per sample. Okay, and so now that we've added 10 microliters of the denaturing buffer plus 10 microliters of the sample, we will incubate at 99°C for 15 minutes to heat and denature the proteins. So I have a program called Boil 99, where if I look at it, it's a 15-minute boil, and then it cools down to 4C. Start that. So while our proteins are denaturing during their boil stage, we'll go ahead and prepare the chamber for running the actual gel. The gel we're going to use today is this mini protein TGX stain-free gel, which allows us to run the gel and then use UV imaging to see the protein bands. There are other types of gel options that mostly use a staining method called Kumasi staining, which require a staining and destaining step. We will use this because it is more efficient for this type of gel running. So in order to set up the gel container, which is here, we'll take the lid, and these are designed to run multiple gels at a time. So we need this option, or this thing called a buffer dam, to fill in for the extra second gel. But we'll open the gel package. The key with setting up these gels is be careful with removing the comb from the gel to not disrupt the wells. So we'll just gently pull it. And then the other key thing is it has a piece of tape on the bottom, this green tape, that you must remove to get good uniform running between the lanes. So once we do that, we will open our gel container here. You'll put the gel on one side and the buffer dam and the other, push them together, and close them like this. This has a red and a black marker on it that correspond to the red and black pins. We'll put it in the front here, and now it's ready to fill with the buffer itself. The buffer uses a tris glycine sodium dodecyl sulfate (SDS) buffer, which you fill the internal well with the gels first, all the way to the top, and then you fill to, this has a line for 2 gels. You can run multiple up to 4 gels, but in this case we'll fill to the 2 gel line, which is 700 milliliters total, and I already have pre-measured that. And so now this is all set up to prepare to load the samples as soon as they're done denaturing. So now that our protein samples have been denatured by boiling at 99°C, we are prepared to load the gel. First, we'll load the ladder. Here we're using a benchmark protein ladder, which, because there's 15 wells and only 7 samples and ladders, I will start in lane 4. We have plenty of wells to load. We load 5 microliters of ladder, and we'll load 10 of samples. When you're loading the SDS-PAGE gel, the gel is very thin, and this outside plastic piece can be used to guide the tip of your pipette. And so for going into well 4, I'll put the pipette tip on this plastic bit and go down, and then slowly pipette my ladder out. Okay. Now that I have my ladder, I will move to loading 10 microliters of each sample. Note that with the 15 well gel, the wells are smaller compared to like a 10 well gel, and so you have to be careful not to cross-contaminate your wells. So pipetting slowly is a key here to not overfill the wells, and to get it to push into the other wells. Now that all the protein samples are loaded, I like to load an extra ladder on the end in case for some reason it does not run uniformly down the gel. So I'll go ahead and load 5 microliters of the ladder, in this case into lane 12. Now that all of the samples and ladders are loaded, making sure that my positive and negative terminals are set up correctly. I'll close the lid, and we have our power source. These SDS-PAGE gels run at roughly 200 volts. So go to the high setting, and in this case 209 is close enough. So now that the gel has run, we let the die front go all the way to the bottom, which in this case took about 20 minutes. We will go ahead and open the cassette. Now this is full buffer, so you can open it up and let it drain out. Then remove your gel. And here you can see the, I don't know if we can get it on the camera out, you can see the die front. Note that the SDS-PAGE gels, they come in this plastic casing, and the gel is on the inside. It requires a tool, a special tool, which I've now covered in suds, to crack open the plastic to access the gel. To do that, it has these arrows that indicate where to crack it open. So you just insert the tool, and you turn it to the side at the plastic arrows, and it cracks open.
Can you move your left arm?
Thanks. I can't crack it that way. Maybe. Insert near the black arrows. Turn it sideways. And then you can move along the edge to pry it open. Note that the SDS-PAGE gels are much more fragile than a DNA gel. And take special care when trying to get them onto the gel imager. I'm not going to remove the case here. Well, I can move it here. It will stick to one side of the plastic, but I'm not going to try to remove it off the second sheet of plastic until I get to the gel imager because they're fragile. We will put this on the gel imager and expose it to UV light for 5 minutes, a 5-minute exposure, to develop the stain, and then you'll be able to see the bands from your protein.